Welcome to our deep dive today.
Yeah,
As medical microbiologists, you're on the front lines of a battle that's getting more intense every year, the fight against bacterial infections.
absolutely.
We're going to take a deep dive into the world of antibiotics,
Yeah.
exploring the challenges, the intricate mechanisms, and hopefully some glimmers of opportunity. We'll be using antibiotics,
Mm.
challenges, mechanisms, opportunities by Walsh and Wencewicz as our guide.
It's a battlefield that's constantly evolving.
Right.
And the enemy, well, they have a knack for adapting.
That's for sure. Almost 15 million people dying each year from infections globally. That's a sobering statistic. Pneumonia, diarrheal diseases, and tuberculosis,
Yeah.
they're still major threats.
Yeah,
And sometimes it feels like we're barely keeping up.
absolutely. And the fact that antimicrobial peptides, nature's own antibiotics, essentially have been around for millennia shows just how long this fight has been going on. Did you know some frog species produce peptides so potent they could rival our most powerful drugs?
It's incredible,
Yeah.
isn't it? It makes you wonder if we've only just scratched the surface of what nature has to offer. And speaking of scratching the surface, let's talk about the history of our synthetic antibiotics. We've come a long way since Prontosil in 1935.
Prontosil was a game changer. It was the first commercially available antibacterial drug and a precursor to the sulfonamides, which inhibit bacterial DNA synthesis. But the story of its discovery is fascinating. It was actually a dye that showed promising results in animal studies, but it wasn't until later that researchers realized it was being metabolized into an active antibacterial compound in the body.
Wow. So it's a reminder that sometimes breakthroughs come from unexpected places.
Absolutely.
But as you said earlier, the bacteria are always adapting, and that brings us to the looming issue of resistance. It's almost like a law of nature, inevitable resistance.
It's a constant arms race. Bacteria, with their sheer numbers and rapid mutation rates, are incredibly efficient at evolving resistance mechanisms. To give you a sense of scale, imagine a population of 100 million bacteria. If just 1 in a million carries a mutation conferring penicillin resistance, you're still left with 100 resistant bacteria ready to take over when penicillin is introduced.
It's a chilling thought, isn't it? It highlights the urgency of finding new antibiotics and, more importantly, understanding resistance mechanisms in depth.
Absolutely. And to make matters more challenging, we have those pesky gram-negative bacteria with their double membrane fortress. It's like trying to break into a bank vault.
You're telling me it feels like every time we find a promising compound, it's 10 times more effective against Staphylococcus aureus than Escherichia coli.
Right.
Why is that outer membrane such a formidable barrier?
It's a complex structure that acts as a selective filter controlling what enters and exits the bacterial cell. Many antibiotics simply can't penetrate it effectively, and even if they do, they often encounter efflux pumps, which are like tiny molecular vacuum cleaners that pump the antibiotic right back out.
So it's not just about finding a molecule that can kill the bacteria, but one that can actually reach its target inside the cell and stay there long enough to do its job.
Precisely. And that's just the beginning. Even when we find an effective antibiotic, the treatment itself presents unique challenges depending on the infection. Take gonorrhea, for example. A single dose can often clear the infection, but compare that to tuberculosis or a chronic leg ulcer, which can require months of antibiotic therapy. It's a completely different ballgame.
And let's not forget about the host's immune system, which plays a crucial role in the success of any treatment.
Absolutely. Immunocompromised patients are at a much higher risk of developing serious infections, and their treatment requires a delicate balancing act. We need to be aggressive in tackling the infection while also protecting the patient from further harm.
It's a constant challenge, weighing the risks and benefits. Speaking of challenges, let's talk about our historical reliance on empiric therapy and broad-spectrum antibiotics. It's like using a sledgehammer to crack a nut.
It's a common practice, especially in urgent situations where we can't wait for culture results. We need to act quickly to save lives. But as you alluded to, there are downsides.
Well, besides the fact that we're essentially blasting the patient's entire microbiome,
Excellent.
it's like we're giving the bacteria a crash course in resistance development. It's no wonder we're seeing resistance spread across multiple species.
You've hit on a crucial point. We're not just treating a single infection, we're interacting with a complex ecosystem. The human microbiome, that vast community of microorganisms living in and on our bodies, plays a crucial role in our health.
So using broad-spectrum antibiotics is a bit like carpet bombing an entire forest to get rid of a few pesky weeds. We might eliminate the immediate threat, but we also inflict significant collateral damage.
Exactly. And that's why there's growing interest in narrow-spectrum antibiotics. It's about precision targeting, minimizing collateral damage and preserving the delicate balance of our microbial ecosystems.
It makes sense. But I imagine developing and using narrow-spectrum antibiotics comes with its own set of challenges.
Indeed, there are economic hurdles to overcome since they target a smaller market. And we also need rapid and accurate diagnostic tests to identify the specific bacteria causing the infection before we can even consider using a narrow-spectrum antibiotic.
So it's a bit of a catch-22. We need better diagnostics to inform the use of narrow-spectrum antibiotics, but we also need the economic incentive to develop those diagnostics in the first place.
It's a complex issue, no doubt, but it's a conversation we need to have. How can we balance effective treatment with minimizing resistance and preserving the microbiome? It's a question every medical microbiologist should be grappling with.
Food for thought, indeed. Now let's shift gears and talk about the antibiotic pipeline. From what I've read in Walsh and Wencewicz, the process of discovering and developing a new antibiotic is a long and arduous journey.
It's a marathon, not a sprint. We're talking about a decade or more and $1 billion to bring a new antibiotic to market. And the success rate, well, let's just say it's not for the faint of heart.
Wow, so talk me through it. Where does it all begin?
It usually starts with a massive screening effort, combing through libraries of synthetic compounds or exploring natural sources like soil bacteria, fungi, even marine organisms.
Ah, the microbial world.
Yeah.
Always full of surprises.
Absolutely.
So once you have your samples, what happens next?
Then comes the high-throughput screening, where researchers test these samples against panels of bacterial pathogens looking for any sign of antibacterial activity.
And if they find a promising hit, a molecule that shows potent activity against, say, a multi-drug resistant strain.
That's when the real work begins. They need to understand its mechanism of action, tweak its structure to optimize its efficacy and safety, and then scale up production for testing in animal models.
So this is where the medicinal chemists really shine, right? They're like molecular architects, designing and building these intricate molecules with the potential to save lives.
It's a meticulous process involving a lot of trial and error, and if they're lucky, they'll identify a lead compound worthy of moving into clinical trials.
And that means?
Human testing. It starts with phase 1 trials, assessing the drug's safety and dosage in healthy volunteers. Then, if everything looks good, it progresses to phase 2, where the drug is tested in patients with specific infections to evaluate its effectiveness. And finally, if it continues to show promise, it moves on to large-scale phase 3 trials, where it's compared to existing standard treatments.
It's a long and winding road with numerous hurdles along the way. But if the drug successfully navigates all those stages, it can finally be submitted for regulatory approval.
Right. Agencies like the FDA scrutinize the data to ensure the drug meets all the necessary safety and efficacy standards before it can be made available to patients.
It's a testament to the dedication and perseverance of countless researchers, scientists, and clinicians who work tirelessly to bring these life-saving drugs to the world.
And it highlights the importance of continued investment and innovation in antibiotic research. We can't afford to let our guard down.
Now, looking back at the history of antibiotic approvals, it's striking to see that there was a 40-year period between 1960 and 2000 when no new classes of antibiotics were approved. It's what Walsh and Wencewicz call the innovation gap.
It was a concerning time for the field. Why do you think that happened?
Well, several factors were at play. The success of earlier antibiotics might have led to a sense of complacency, a belief that we had conquered bacterial infections.
And there were also limitations in our discovery methods. We were relying heavily on screening natural sources, and those were starting to yield diminishing returns.
So we were essentially hitting a wall with the conventional approaches, but thankfully we've seen a resurgence in antibiotic approvals since 2000.
Yes, advances in genomics, synthetic chemistry, and drug screening technologies have opened up new avenues for discovery. We're beginning to understand bacterial physiology at a much deeper level, identifying new targets and designing more sophisticated molecules.
It's encouraging to see this renewed focus on antibiotic research. But let's be honest, the threat of resistance is still looming large, and it's getting worse.
You're absolutely right. We're seeing alarming trends of rising resistance rates and a decline in the discovery of new antibiotics. It's a recipe for disaster.
Walsh and Wencewicz don't mince words. They paint a stark picture of the potential consequences, using terms like flesh-eating bacteria and killer E. coli.
It's not hyperbole. We're seeing infections caused by bacteria that are resistant to nearly all available antibiotics. And these infections are becoming increasingly common not just in hospitals, but in the community as well.
It's a sobering reality, and one that we as medical microbiologists need to confront head-on. But where do we even begin?
Well, the first step is recognizing the multifaceted nature of the problem. It's not just about bacteria evolving. Our own actions are playing a significant role.
You're talking about the overuse and misuse of antibiotics, both in human and animal medicine.
Exactly. The widespread use of antibiotics in agriculture, where they're often used as growth promoters in livestock, is a major contributor to the problem.
And then there's the issue of inappropriate prescribing. Too often, antibiotics are prescribed for viral infections, which they have no effect on. It's like trying to put out a fire with a garden hose.
And even when antibiotics are prescribed appropriately, patients often don't complete their full course of treatment, allowing resistant bacteria to survive and multiply. It's a vicious cycle.
So it's a complex web of interconnected factors driving this resistance crisis. But it's not all doom and gloom, is it? Walsh and Wencewicz do offer some glimmers of hope.
Absolutely. There are a number of promising avenues for addressing this challenge, and I'm curious what stands out to you as particularly promising.
Well, the idea of exploring underexplored targets within bacteria is intriguing. Instead of going after the same old targets that bacteria have already developed resistance to, we need to find their Achilles heel, something they haven't encountered before.
It's about outsmarting the bacteria, finding their weaknesses, and exploiting them before they can adapt. And thankfully, advances in genomics and proteomics are giving us unprecedented insights into bacterial physiology. We're starting to unravel the intricate workings of these tiny cells, identifying potential targets we never knew existed.
It's like having a detailed blueprint of the enemy's fortress. And with that knowledge, we can design more targeted weapons drugs that disrupt essential bacterial processes without causing widespread damage to the host.
Precisely. But it's not just about finding new targets. We also need to think about new sources of antibiotics. We've traditionally focused on soil bacteria and fungi, but there's a whole world of untapped potential out there.
You're talking about exploring the oceans, insects, even plants. It's amazing to think that the solution to this global crisis could be hiding in the most unexpected places.
Nature is a master chemist, and we've only just begun to tap into its vast arsenal of bioactive compounds. For example, researchers have discovered novel antibiotics from deep-sea sponges, the venom of poisonous spiders, even the saliva of Komodo dragons.
It's like a real-life treasure hunt searching for these molecular gems with the power to combat resistant bacteria.
And it's not just about discovering new antibiotics. We also need to get smarter about how we use the ones we already have.
You're talking about antibiotic stewardship, right? Using antibiotics responsibly and only when necessary.
Exactly. We need to educate both healthcare providers and the public about the importance of appropriate antibiotic use. This includes prescribing antibiotics only when they're truly needed, ensuring patients complete their full course of treatment, and avoiding the use of antibiotics for viral infections.
It's about changing behaviors and promoting a culture of responsible antibiotic use.
And it's about investing in rapid diagnostic tests that can quickly identify the specific bacteria causing an infection, allowing for more targeted treatment with narrow-spectrum antibiotics.
So it's a multi-pronged approach combining scientific innovation with behavioral changes in policy interventions.
It's a complex challenge, but one that we must overcome. The stakes are simply too high.
Well said. Now, before we move on to part 2 of our deep dive, let's take a moment to test your knowledge with a few exam questions.
Sounds like a good challenge. Let's see what you've learned so far.
All right, get ready. Here's your first question. Why is antibiotic resistance considered an inevitable consequence of antibiotic use? Take your time. Think it through.
This is a fundamental concept, but it's worth revisiting. It really boils down to the nature of bacteria themselves. They're incredibly prolific, huge populations, and rapid replication rates.
Right.
That means a lot of opportunities for mutations to arise.
So even if the chance of a resistance mutation is low, with billions of bacteria multiplying, it's bound to happen eventually.
Exactly. It's like playing the lottery. The more tickets you buy, the higher your chances of winning. And in this case, the prize is survival in the face of an antibiotic attack.
And those resistant bacteria can then multiply and pass on their resistance genes to others.
Right.
Even across different species.
Right. That's the scary part. It's not just about individual bacteria developing resistance. It's the potential for those resistance genes to spread like wildfire through the bacterial community. Horizontal gene transfer, they call it.
Wow. It's like a bacterial arms race, and we're struggling to keep up.
It is.
All right, ready for question number 2?
Absolutely. Let's hear it.
Okay, here it is. What structural feature makes gram-negative bacteria more challenging to treat with antibiotics compared to gram-positive bacteria? Think back to our discussion about their outer membrane.
Ah, yes. That double membrane is a real headache. It's like a fortress wall. with selective entry points and efflux pumps that can kick antibiotics right back out.
So even if an antibiotic can get through the outer membrane, it still has to contend with those efflux pumps.
Exactly.
It's like trying to sneak into a concert and being thrown out by security.
A very fitting analogy, and this is why screening for new antibiotics often yields far more hits against gram-positive bacteria like Staphylococcus aureus compared to gram-negative foes like E. coli.
It really highlights the need for new strategies to overcome this gram-negative barrier. Maybe we need to design antibiotics that can bypass those efflux pumps or disrupt the outer membrane structure itself.
There's a lot of exciting research in that area looking at ways to enhance antibiotic uptake, inhibit efflux pumps, or even use Trojan horse strategies to sneak antibiotics into the cell.
Now that's thinking outside the box.
Yeah.
Okay, moving on to question number 3. This one delves a bit into history. What is the innovation gap in antibiotic development, and what factors contributed to it?
The innovation gap refers to that rather worrying period between 1960 and 2000 when no new classes of antibiotics were approved for clinical use.
Right. It's like the antibiotic pipeline went dry for 4 decades.
It did.
Why do you think that happened?
There were several contributing factors. Some experts believe there was a sense of complacency, a feeling that we had conquered infectious diseases. After all, we had penicillin, streptomycin, and tetracycline. It felt like we had all the weapons we needed.
A dangerous assumption, as we now know.
It was indeed. What else contributed to this gap?
Well, yeah.
Traditional discovery methods relying primarily on screening natural sources were starting to hit their limits.
Okay.
We were finding fewer and fewer novel compounds, and the economic incentives for antibiotic development weren't as strong as for other therapeutic areas.
So a combination of scientific limitations and market forces essentially stifled innovation.
Exactly. It was a perfect storm that created this innovation gap, and it took a while for the urgency of the situation to be fully recognized.
Thankfully, we've seen a resurgence in antibiotic approvals since 2000. But the need for continued innovation remains paramount.
Absolutely. We can't afford to become complacent again. The bacteria are constantly evolving, and we need to stay one step ahead.
Okay, ready for question number 4?
Bring it on.
All right, here it is. Why is the reliance on broad-spectrum antibiotics a double-edged sword in the fight against bacterial infections? Think about the potential benefits and drawbacks.
Broad-spectrum antibiotics can be incredibly valuable, especially when we're dealing with a critically ill patient and can't wait for culture results. They provide a broad safety net covering a wide range of potential pathogens.
Right.
But there's a downside.
It's like using a shotgun instead of a sniper rifle.
Yeah.
You might hit your target, but you're also likely to cause a lot of collateral damage.
Exactly. By targeting such a broad range of bacteria, we risk disrupting the delicate balance of the microbiome, wiping out beneficial bacteria alongside the harmful ones.
And that can lead to all sorts of unintended consequences, from digestive issues to opportunistic infections,
Absolutely.
even long-term impacts on immune function.
Precisely. And perhaps the biggest concern is that widespread use of broad-spectrum antibiotics accelerates the development of resistance. It's like we're giving the bacteria a crash course in survival skills.
So it's a delicate balancing act.
Yeah.
We need to use broad-spectrum antibiotics judiciously, only when absolutely necessary, and strive to transition to narrow-spectrum agents whenever possible.
Absolutely. Now ready for the final challenge. Okay, last question. Name 2 potential strategies for addressing the growing threat of antibiotic resistance. Think big picture beyond specific antibiotics.
So this is about more than just finding new drugs.
Yes. This calls for a systemic approach, not just focusing on individual drugs. I'd say 2 critical strategies are investing in rapid diagnostic tests to enable more targeted treatment and promoting antibiotic stewardship to reduce unnecessary use.
Those are both excellent choices. Rapid diagnostics could revolutionize how we approach infections. Imagine being able to quickly and accurately identify the specific bacteria causing an infection right there at the point of care.
It would allow us to tailor treatment to the individual patient using the most appropriate antibiotic at the right dose, minimizing the risk of resistance development and side effects.
And antibiotic stewardship is all about changing prescribing behaviors, educating patients, and implementing policies that promote responsible antibiotic use.
It's a multifaceted approach that requires collaboration among healthcare providers, policymakers, and the public. We need to work together to preserve the effectiveness of these precious drugs for future generations.
Well said. It's clear that the fight against antibiotic resistance is a complex and multifaceted one requiring a multi-pronged approach. Now, in part 3 of our deep dive, we'll delve into those exam questions in more detail, exploring the reasoning behind the answers and providing additional insights.
I'm looking forward to unpacking these concepts further and exploring some of the cutting-edge research that's giving us hope in this battle against resistance.
Welcome back to the final part of our deep dive into the world of antibiotics. Are you ready to unpack those exam questions and see how well you did?
I am. Let's dive in.
Okay, great. So let's revisit that first question. Why is antibiotic resistance considered an inevitable consequence of antibiotic use?
Well, it really comes down to the sheer scale of bacterial populations and their incredible ability to adapt. Bacteria reproduce rapidly, and with each division, there's a chance for mutations to arise. Even if a single mutation conferring resistance is rare, when you're dealing with trillions of bacteria, those rare events become almost certainties.
So it's a numbers game.
Yeah.
The more bacteria there are, the higher the chances of resistance popping up.
Exactly. And once a resistant strain emerges, it can quickly spread through horizontal gene transfer, sharing those resistance genes with other bacteria. It's a constant evolutionary arms race, and the bacteria are remarkably adept at staying ahead.
That's kind of a sobering thought when you think about it.
Aye.
All right, let's move on to question number 2. What structural feature makes gram-negative bacteria more challenging to treat with antibiotics compared to gram-positive bacteria?
This one takes us back to the basics of bacterial structure. Gram-negative bacteria have that formidable outer membrane, a double-layered barrier that many antibiotics struggle to penetrate.
Right.
It's like a fortress wall protecting the bacteria from our antibiotic arsenal.
And even if an antibiotic manages to get past that outer membrane, it still has to contend with those pesky efflux pumps.
Right.
Those molecular vacuum cleaners that pump the antibiotic right back out of the cell.
Precisely. It's a multi-layered defense system that makes treating gram-negative infections particularly challenging. No wonder we see such a disparity in the effectiveness of many antibiotics against gram-positive versus gram-negative bacteria.
Yeah, it really is remarkable. All right, on to question number 3. What is the innovation gap in antibiotic development, and what factors contributed to it?
This is a fascinating, albeit somewhat disheartening chapter in the history of antibiotic research. The innovation gap refers to the period between 1960 and 2000 when no new classes of antibiotics were approved for clinical use.
Yeah, it's like the antibiotic pipeline went dry for 4 decades. Why do you think that happened?
A combination of factors converged to create this perfect storm. After the golden age of antibiotic discovery in the mid-20th century, there was a sense of complacency, a belief that we had conquered infectious diseases.
A dangerous assumption, as we now know.
Indeed. And at the same time, the traditional methods for discovering new antibiotics, primarily through screening natural sources, were yielding diminishing returns. We were finding fewer and fewer novel compounds.
So a combination of scientific limitations and perhaps a bit of overconfidence led to this stagnation.
Exactly. And let's not forget about the economic factors. Antibiotic development is a risky and expensive endeavor, and the financial incentives weren't as strong as for other therapeutic areas.
Right, so it was a confluence of factors that ultimately led to this innovation gap. Thankfully, we've seen a resurgence in antibiotic approval since 2000, but the need for continued innovation remains critical. All right, 2 more questions to go. Ready for number 4?
Absolutely.
Here it is. Why is the reliance on broad-spectrum antibiotics a double-edged sword in the fight against bacterial infections?
This is a question that really gets to the heart of antibiotic stewardship. Broad-spectrum antibiotics, those that target a wide range of bacteria, can be lifesavers, especially in emergency situations where we don't have time to identify the specific culprit. But their widespread use comes at a cost.
Right. It's like using a sledgehammer when a scalpel would suffice. You might eliminate the immediate threat, but you also risk causing collateral damage.
Precisely. Broad-spectrum antibiotics don't discriminate between good and bad bacteria. They disrupt the delicate balance of the microbiome, wiping out beneficial bacteria alongside the pathogens.
And that can lead to all sorts of unintended consequences, from digestive issues to opportunistic infections, even long-term impacts on immune function.
And perhaps the most concerning consequence is the acceleration of antibiotic resistance. By exposing a broad range of bacteria to these drugs, we're essentially selecting for resistant strains.
So it's a classic case of too much of a good thing. Broad-spectrum antibiotics have their place, but we need to use them judiciously and strive to transition to narrow-spectrum agents whenever possible.
Absolutely. It's about finding that balance between effectiveness and minimizing unintended consequences.
All right, ready for the final challenge?
Bring it on.
Okay, last question. Name 2 potential strategies for addressing the growing threat of antibiotic resistance. Think big picture beyond specific antibiotics.
This is where we need to shift our focus from individual drugs to a more holistic systems-level approach. I'd say two key strategies are investing in rapid diagnostic tests and promoting antibiotic stewardship.
Those are both excellent choices. Rapid diagnostics could revolutionize how we approach infectious diseases, allowing us to quickly and accurately identify the specific bacteria causing an infection.
Imagine a world where we can get a definitive diagnosis within minutes, right there in the doctor's office or even at home. It would allow us to tailor treatment to the individual patient using the most appropriate antibiotic at the right dose.
And antibiotic stewardship is all about promoting responsible antibiotic use across all sectors, from human and animal medicine to agriculture and the environment.
It's about educating healthcare providers and the public implementing policies that restrict the use of antibiotics in livestock and developing new guidelines for appropriate prescribing.
Right. It's a multifaceted effort that requires a coordinated global response.
Yeah.
We need to work together to protect these precious drugs for future generations. Well, that wraps up our deep dive into the world of antibiotics. We've covered a lot of ground, from the history of antibiotic discovery to the challenges of resistance and the promising opportunities for the future. It's clear that medical microbiologists like you, our listener, are at the forefront of this battle. Your expertise and dedication are crucial in shaping the future of infectious disease treatment. What are your final thoughts?
I completely agree. It's been an incredible journey exploring the intricacies of antibiotic action, the complexities of bacterial resistance, and the urgent need for new solutions. But it's also been inspiring to see the incredible work being done by researchers, clinicians, and policymakers around the world to combat this global threat.
And remember, you're not alone. There's a whole community of dedicated individuals working tirelessly to address this challenge. So as we conclude this episode, we leave you with a challenge. What role will you play in safeguarding the future of antibiotics and protecting human health from the threat of untreatable infections? The answer lies within each of us. Stay curious, stay informed, stay engaged, and together let's make a difference.